Hey there, my name is Hayden Hyatt. I am a PhD student here at Auburn University, and today I'm going to be walking with you the process of Western blotting. So Western blotting is how we actually measure proteins in our samples here at Auburn and all over the world really. It's a widely used process. But first off, what we start with is we start with our tissue of interest, so perhaps liver or skeletal muscle. And we're going to homogenize it so we get a nice ubiquitous blend or a very thorough blend of proteins of that sample. And then after that, we're going to be doing several other steps where we actually measure these proteins, and I'll walk you through that. So after we have all of our proteins isolated out from our sample or our tissues, the next process that we're going to do is called gel electrophoresis. And so what this is this is essentially a gelatin structure that we've made, and we put them into these glass cassettes. And we're going to basically separate our protein by molecular weight. So we're going to separate it basically by how big they are. And so the way to think about this really is if you were to have a bamboo forest and you had pandas running through this bamboo forest, the bigger pandas aren't going to quite make it as deep into the forest as the smaller ones would. And so the way that we accomplish this is we'll actually put this in this box here, and we're going to run electricity through it, and so that's going to polarize these proteins and move them through this gel so that we can separate them. And then after that, we're going to be going on to the next phase where we'll actually set them up and we'll be able to quantify how much protein is actually in those tissue samples. And so now we enter our last stages of performing the Western blot process, and this is where we actually get the data from it. So it's obviously a very long process, but so at this point, we're going to have taken the protein that's in that gel, and we will use electricity to then transfer it onto a membrane or a sort of type of paper that will then contain all the proteins that have been separated out on that. At that point, we're going to add antibodies to it so that we can actually quantify how much protein is on it. So, such as catalase, this is what would happen. We would take a mouse, and we would isolate that protein catalase from that mouse. Then we would take that protein and we would actually inject it into a chicken, and that chicken would produce antibodies to it. So basically these antibodies are just substances that are going to be produced by the person or the chicken that are going to recognize that foreign object of catalase, and then will be able to bind to it and tag to that protein. And so this is kind of like how if you were to get the flu, you wouldn't get the flu again because you have antibodies against the flu virus that would be able to recognize it and stop the flu from causing its harmful effects again. So that's our what's called our primary antibody. We would then take this primary antibody and put it into a rabbit, and that rabbit would recognize that antibody as foreign, and so it would produce antibodies against that antibodies. And so then what would happen is we would put our primary antibody on that Western blot, and then we would put, and that would tag to the catalase that's in that protein that we measured. And then we would also put that secondary antibody, and this secondary antibody has also been equipped with a little phosphorus sensor, essentially, and so when we add chemicals to it, it's going to cause it to release light. And so when that light is released, if you can come here, you can see, we can actually quantify it with the picture, and we would be able to tell how much protein is actually equipped to that membrane. And so what you can see here, this is an example of a study that we did measuring sedentary individuals versus people that exercise over a certain period of time. And we can actually look at the amount of antioxidants, the differences between these individuals. And so you can see here some of these lighter bands actually represent the sedentary individuals. They didn't have as many antioxidants. And these darker bands represent the exercise individuals, and these are because they had more antioxidants than sedentary people. And so then we can actually quantify that, and it gives us a way to actually measure how much protein was actually in the muscle for these individuals.